This is a brief introduction to some of the things you want to know about before you start reading the last paper that we'll discuss this semester. Here are the things that I'll cover in this lecture. We'll talk about a brief history of coronavirus epidemics and pandemics, take a look at China's limestone cave systems. Then we'll talk in depth about the virology of coronaviruses. I'll also discuss a number of the key scientific experiments that the scientists perform in this paper. In the virology section, I'll discuss how we use cells and cell culture to culture human viruses, talk about phylogenetic analysis, and finally, talk about immunofluorescence microscopy. First, I'd like to give you a brief history of coronavirus pandemics. So, in November 2002, a farmer in Guangdong province, China, was admitted to a hospital and later died of a severe respiratory illness, of which nobody could identify a cause. There were several more cases of this severe respiratory illness around the region for several months, and in December 2002, the World Health Organization identified the cause, which was a new type of coronavirus that they termed severe acute respiratory syndrome coronavirus, or SARS-CoV. SARS-CoV caused severe respiratory illness, so people would get really sick and had nearly a 10% fatality rate. This virus spread around the world in part because of a number of aspects of our modern society, where people traveled frequently by airplane from Southeast Asia, where the epidemic started, around the world. We had a number of cases of spreading the disease to hospital workers and other individuals around the world throughout the next several months. Thankfully, due to a good public health response around the world and some aspects of this virus, for example, people became sick very quickly after acquiring the virus. We were able to identify cases and prevent the spread of this disease around the world. At the end of the pandemic, only 8,437 people had become infected, although 813 of them had died of the disease. Then in June 2012, a man in Saudi Arabia was diagnosed with acute pneumonia, again of unknown cause. And then there were cases that occurred sporadically around the Middle East for several months. In November 2012, virologists in Egypt and the Netherlands identified another new coronavirus. In this case, what they termed Middle East Respiratory Syndrome coronavirus, or MERS-CoV. So MERS-CoV also causes a severe respiratory illness. It has a high fatality rate. Luckily, this virus doesn't seem to be very good at transmitting person to person, but it does seem to be reasonably good at transmitting from camels, which seem to be its primary host, to humans. And since camels are a domestic animal in many parts of the Middle East, we've had several occurrences where people have been infected with MERS coronavirus from domestic camels. So these cases were mostly sporadic until May 2015, when a new strain, another strain of MERS coronavirus, caused a significant outbreak in South Korea, where there were 186 cases and 36 deaths from this one outbreak. So, if you weren't seeing a pattern in the data, in December 2019, there were several cases of a severe pneumonia that were linked to a seafood market in Hubei province, China, near the city of Wuhan. An earlier case was later identified that had no connection to the market, so that marketplace does not seem to be the start of this disease. However, there were cases spread around China for a number of months, and by December 2018, DNA sequencing had identified a new coronavirus in the patients from Wuhan, China. And that virus was very closely related to the original SARS-CoV. By February 2020, the WHO had named the disease caused by SARS-CoV-2 COVID-19. In January and February 2020, we had the first deaths in Europe and the United States, and for a number of reasons. Perhaps because of some of the aspects of SARS-CoV-2 biology, especially the fact that this virus seems to be present asymptomatically in individuals either before they become ill or in individuals who never become ill, who can nevertheless spread the disease to others without them being aware that they're sick. Or the ineffectual public health response of governments around the world, or the widespread misinformation that we see in the world today. This has become one of the most severe pandemics in human history. We have over 45 million cases, and as of when I printed this slide, over 1.18 million deaths. Another thing that you should understand about coronaviruses is the geology and ecology of Southeast Asia. So, Southeast China has the largest deposit of karst in the world. So karsts are rock formations that are formed when soluble minerals in the rock are dissolved by flowing water. So this creates these incredible geological formations, including these cliffs that are found in Wulingyong National Park, which is about a 5-hour drive from Wuhan, China. And it also creates some enormous cave and mountain systems in China, all over Southeast Asia. These caves are home to many animals, including many species of bats. So bats are in the mammalian order Chiroptera, and we know that bats are mammals, but they are clearly much different from many of the other mammals on the planet. They can fly, and they have some interesting biology. So it's the order Chiroptera is the second most diverse in the mammalian group. There's over 1,400 species. That's 22% of all known mammal species. And bats harbor many viruses. So bat viruses that infect humans include Nipah virus, rabies virus, quite likely Ebola virus, although we don't have definitive data about that yet, and many types of coronaviruses. So these horseshoe bats are just one of the many species that live in the limestone caves in Southeast Asia. So spillover of diseases from animals to humans are called zoonotic diseases. So you recall from our early discussions of bacteriophages that many viruses and other pathogens are well adapted for a specific host. We call this viral tropism. However, some viruses are capable of cross-species transmission. This event is uncommon. Most viruses are well adapted for a specific host. But as we saw in our previous paper, if you test enough viruses, they can infect multiple different hosts. You're going to increase the probability of one of those rare variants in a population of viruses jumping from host to host if there's more exposure of those viruses in the animals that they are reservoired in to people. Right? So we've seen a number of instances in our modern world where viruses and other pathogens have spread from animals, both domestic and wild, into people, both as the result of increasing contact between wildlife and human populations as our populations over the globe expand, and also as the result of some of our modern farming practices. Going forward, we're going to have to think very carefully about how we manage our interactions with both wild and domestic animals in order to prevent these pandemic diseases. So next I want to talk about coronavirus virology. So SARS-CoV-2 is extremely similar to SARS coronavirus. And so many of the things that I will tell you about SARS-CoV-2 and SARS coronavirus are very similar, if not quite closely the same. So coronaviruses are an enveloped RNA virus. Right? So they have a phospholipid bilayer surrounding their capsid, and they have an RNA genome. Coronaviruses are called coronaviruses because they have this crown-like appearance when you look at them under an electron micrograph. That's because they have this spherical capsid surrounded by the phospholipid bilayer envelope. And then they have a number of spike proteins that stick out from the capsid and the membrane envelope. So there are 7 coronaviruses known to infect humans. So 4 of them, called 229E, NL63, OC43, and HKU1, these are all viruses that cause what we usually call the common cold. They cause mild upper respiratory infections. We talked about MERS coronaviruses that cause severe respiratory infections, but have little person-to-person transmission. We also talked about SARS coronavirus, which is the subject of the paper that you'll be reading, that causes severe respiratory illness, and we discussed the outbreak in November 2002. And then finally, the SARS-CoV-2 is the cause of the current COVID-19 pandemic. So SARS-CoV-2 and SARS-CoV both replicate in much the same way. So you'll remember our discussions of the lytic and lysogenic cycle of bacteriophages. Remember that viruses can't reproduce on their own, and so they need to get into a host by attaching to the host cell, penetrating the host cell, and then getting the host to biosynthesize or reproduce all of their virus parts that are then assembled into new viruses and released, in the case of bacteriophages, by lysis from the host cell. So the reproduction of SARS-CoV, a mammalian virus, goes a little bit similar. So SARS-CoV attaches to a susceptible host cell by binding to a receptor protein on the mammalian cell surface, which is called ACE2. So ACE2 is the receptor that's present on the mammalian cell surface. The viruses bind to ACE2 through an interaction with the protein spike called S, which protrudes from the virus capsid. Right? So S on the virus sticks to ACE2 on the host cell, and that induces the host cell to go through a process called endocytosis, which is where mammalian cells engulf particles from the outside in a membrane component or in a membrane compartment, and then bring those particles inside the cell. So the virus is kind of tricking the cell into gaining entry into the inside. So once the virus is on the inside of the cell, it is unpackaged, and the viral genome is released into the cell. So the SARS-CoV genome is RNA, and so RNA can be translated into proteins inside the cell, and so that genome is used to make all of the SARS-CoV proteins. One of the proteins that is made is the SARS RNA-dependent RNA polymerase. Because the next thing that has to happen is that the virus's genome needs to be reproduced, and the enzyme that does that is called RNA-dependent RNA polymerase. Once those genomes are replicated and all the new virus proteins are made, then new viruses assemble inside the cell. Then, once those new virions are assembled, they induce the cell to exocytose, or engulf them in a phospholipid bilayer envelope, and eject them from the host cell. Right. So we learned in the bacteriophage lytic reproduction cycle that the a host cell is destroyed as a result of lysis. That is not the case for many mammalian viruses, including SARS-CoV. However, the reproduction of the virus inside the cell, while it doesn't kill all of the cells that they reproduce in, so the virus can exit through exocytosis without completely destroying the cell, it does cause cell damage, and many of the cells do eventually die. So we mentioned the RNA-dependent RNA polymerase, and so this is a little bit of a preview from Unit 5, where we'll talk about how genes work in cells. And so in cells, all the genes are encoded in the DNA molecule. But proteins are the thing that do most of the work inside of the cell. And so in order for those genes in the DNA to be turned into proteins, first a copy of the gene is made in mRNA, and that process of making the RNA is called transcription. And then the mRNA is translated into a protein. Viruses, so that process happens on all cells, but viruses are a little bit different. So some viruses don't have DNA genomes, they have RNA genomes. And in order for their RNA to be translated into proteins, that RNA just is read by host cell ribosomes. But they also need a special enzyme to reproduce their RNA genome, or replicate their RNA genomes, and that enzyme is called an RNA-dependent RNA polymerase, because it replicates RNA from an RNA template. Next preview of Unit 5 that you'll need to know about to understand this paper is what is an ORF, or an open reading frame. So, as we discussed, genes are pieces of genetic information inside of genomes. Right? So those genes are encoded in the DNA, and in order to be turned into proteins, a copy needs to be transcribed into a messenger RNA. And then inside of the gene, each 3 nucleotide sequence is read by the ribosome. So these are called codons. And each one of those 3 nucleotide codons specifies one of the 20 amino acids that makes up proteins. And so the codons are decoded, and the protein is made during the process of translation. So if you look at a gene inside of a DNA genome, you can see several parts of the gene that are important for that gene working at the right time and place. So at the beginning and end of the gene, there is a promoter sequence and a terminator sequence. And those are the sequences that direct the RNA polymerase that does the transcribing of the gene into mRNA, where to start and stop. Right? So that the gene can be copied into mRNA at the right time. Now, in the middle of that sequence is the open reading frame. The open reading frame contains the start sequence for translation, the stop sequence for translation, and all of the codons that specify the amino acids that go into making the protein. So if you look at the genome of a virus like SARS coronavirus, you'll see a number of open reading frames for each one of the proteins that are made to make new virus particles. So these lines show the location of these genes in the SARS-CoV and MERS-CoV genome. So we see these first 2 open reading frames, ORF 1A and 1B, and those are protein coding regions that code for a number of proteins, including the RNA-dependent RNA polymerase and some other genes that are necessary for virus genome replication. Then we see in this second ORF, that is the open reading frame that codes for the protein S, or the spike protein that's necessary for attaching to ACE2. Then we see a number of additional open reading frames that encode other genes that make up the coronavirus virion. So each one of these open reading frames codes for a different protein that is important for the virus replication. One thing that this paper does discuss is genetic recombination in viruses. So recombination is a process where nucleic acids that have similar sequences get switched or shuffled. And so this happens in a number of different types of biological organisms. It happens during when DNA is repaired when it's damaged inside of cells. And it also happens when our cells go through meiosis as a way to increase genetic diversity. Right? And this is a figure that shows how that process of recombination happens for mammalian genomes. Right? For viruses, the recombination happens between two sequences that are similar. And it can only happen if two viruses with similar but distinct sequences happen to infect the same cell. So those two virus genomes have to be in the same physical space in order for this crossing over of the chromosomes to happen and the parts of the DNA genomes to get shuffled. So in this paper, the scientists isolate a number of new coronaviruses and look at their gene sequences, both the nucleic acid and the protein. But we also know that we study viruses by growing them in the lab. Right? So our bacteriophage viruses need a bacteria host in order to reproduce. And so in lab, we use Microbacterium floriorum, or in the previous paper, we saw the use of the Mycobacterium smegmatis host, or in the first paper, the Paenibacillus larvae. So if we want to study human viruses or viruses that reproduce in mammalian cells, we need some cells to grow those viruses in. So we grow human viruses by using human cell lines. Right? So we have specific human cells that we're able to grow in lab. One example of these are HeLa cells that were cultured from tissue taken from a cancerous tumor from a woman named Henrietta Lacks in 1951. These cells have been grown in labs around the world and used in a number of experiments that have provided critical breakthroughs in our understanding of viruses and cancer and a number of other processes. Right? But these cells were also taken from Henrietta Lacks, who was African American, without her consent. And there are a lot of really interesting ethical questions around the use of these cells in labs around the world. And you can read about that in Chapter 6.3 in OpenStax Microbiology. Right? However, growing viruses in cell culture is a lot like growing them in our bacteria cells. In fact, this individual in the picture is holding up a plaque assay that they did with influenza virus on this plate. You can see the plaques that are the sites of influenza virus infections on the cells that were growing at the bottom of this dish. But in this case, the cells grow in a single layer on the bottom of the dish. Human cells are a little bit harder to grow than bacteria cells. And so they only grow on a single layer on the bottom of the dish. And so you can't really see them like you see the turbid growth for bacteria. And so instead they use a strong dye, that purple dye, that stains each one of the cells when they are alive. And if those cells are dead, then you see the loss of that purple dye. And that is the visible plaque. So when we grow viruses in a laboratory condition using cell lines, we can do a number of experiments to learn about how the viruses reproduce. One of the things that we do is make recombinant virus vectors. So remember, the SARS-CoV virus enters the host cell as a whole virion and then is unpackaged inside the cell. But our bacteriophages that we work with, they reproduce. The virus stays mostly outside the cell and it just injects its DNA. And so that genetic information has everything that needs to that the cell needs to make new viruses. So we can make viruses from recombined genetic parts by using bacteria artificial chromosomes, or BACs, and those are large circular pieces of DNA. Another word for that is plasmids, that we can copy, or what we call in molecular biology, clone those genes from viruses into. So we can make an artificial piece of DNA that has all of the virus genes that we want to make new viruses on it. And we can take these plasmids and reproduce viruses by growing them in cells. Now, this technique, a number of people have raised concerns that we might be able to generate new and dangerous viruses by using this technique. But for the most part, it's actually, in practice, really hard to get good viruses by cloning together different parts from viruses. So we have been, even if people were trying, it would be very difficult for somebody to make a superbug from this technique. Usually you're lucky for your experiment to work at all, and also you're making these viruses reproduce in cells and not in actual people. However, this can be really useful in telling us what virus genes can and can't do. So when you read this paper, think about some of the following questions. First, how did the scientists collect the different viruses that they examined? What genes do the scientists look at to determine if the bat coronaviruses are similar to the SARS isolates? Then, what did the authors learn from comparing those gene and protein sequences from the virus? You also want to make a distinction between the ways we learn about viruses from looking at their gene sequences and the ways we learn about viruses from working with them in laboratory experiments, growing them in culture. So think about the different things that the authors learn from those in vitro cell culture experiments. Finally, what cells do the authors use as hosts for their viruses in this paper? So one of the things that the scientists do that's really important for this paper is they make a lot of phylogenetic trees. Right? So phylogenetic trees are a graph that we use to visualize evolutionary relationships. When we look at a phylogenetic tree, we usually only know things about the individual species or strains that we see in current day. So we know that these strains have an evolutionary relationship, right? But we need to figure out that relationship by making some inferences based on the structures that these species have. Right? So we can create a phylogenetic tree, and we'll talk about how we do this. And when we do that, we can map these evolutionary relationships. So a branch point is a point on the graph where two lineages or two different strains or species diverged. And so this is really showing an event that happened in evolutionary history. We don't have a direct observation of this event happening, right? But there was some change in this population of organisms where one group of organisms developed and behaved one way, and another group of organisms developed and behaved another way. The root of the tree is a common ancestor of all of the organisms that are displayed on the graph. So, how do we build evidence for the existence of these evolutionary relationships if we only see the organisms as they exist today? So one of the ways that we do this is by looking at homologous structures. So homologous structures are structures that are common features that are shared between organisms that have evolutionary relationships. For example, in this phylogenetic tree, where we compare a number of animals, lamprey, antelope, bald eagle, alligator, and sea bass, we can look at a number of their structures, many of which are found in all of the organisms that are shown here, and some of which are only found in certain groups. So all of the organisms on this tree have a structure called a notochord, which is that bundle of nerve tissue that runs down your spinal column. But if we look at other structures that are shared by almost all of the organisms on this tree, for example, the jaw, that is missing in the organism called the lamprey. So there was some event in evolutionary history where one group of organisms developed a jaw, while the group of organisms that include the modern-day lamprey developed without that structure. We then see that many of these organisms have structures called lungs, so they have some sort of evolutionary relationship, some sort of shared heritage, whereas fish did not have lungs. And so there must have been an event in evolutionary history where all of these jawed organisms, some developed a lung or a lung-like structure, and fish did not. We can then differentiate the structures such as the gizzard, which is found in both reptiles and in birds, or also hair, which is found only in mammals, in order to define some of the things that are shared by certain groups of organisms, right? But not by others, and infer that there must have been a change in these populations of organisms in the past that caused these distinct groups to be formed. So we can do this homologous structure analysis using physiological structures that we can see, like bones and organs. And we can also look at homologous molecular structures, such as genes. So we can look at sequences of genes, such as these that are shown in the important genes for the ribosome, which is a complex that's responsible for making proteins in all cells. You can see that these structures have some similar sequences between all of the very different organisms that are shown on this figure. For example, we have Mycobacterium sphygmatis, Homo sapiens, Leishmaniasis trypanosoma, which is a eukaryotic parasite. But there are also some distinct features, those structures, those places where the structure has changed during evolutionary history. So if we wanted to create a phylogenetic tree using these sequences, we could take a group of species that we see today and look at some of the common features of their genes, and then look at some of the individual sequence differences. So we have a group here of 4 different species, and then one distant relative that we call the outgroup. So we can create a phylogenetic tree that best represents the fewest number of evolutionary changes that would have needed to happen to generate all of these homologous structures as they exist today. So take a moment here and think about this problem. Which one of these phylogenetic trees represents the most probable development of the different genetic differences that we see in these organisms on the graph? Is it A or is it B? You can pause the video here and think about this question. So if we look at the structures here, we can see that while the distant evolutionary relative has an A at position 3, species 1 and 2 both have a C. So it's possible that at some point in history, while all of these organisms had an A at position 3, the group of organisms that eventually became species 1 had a to C change at that DNA nucleotide. And then later in evolutionary history, another group of organisms that includes species 2 also had the exact same change. However, another possible explanation is that one group of organisms that went on to include both species 1 and species 2 had this A to C change at the same time. And then some additional evolutionary change happened later in time that led to the differentiation of species 1 from species 2. So note that A requires the exact same change to happen twice in evolutionary history, whereas B describes the single change happening in one common event. So B is going to be more likely because it explains this single pattern that we see in the data with one explanation rather than two. Now, things don't always happen in the simplest way possible, but generally the simplest explanation is the best, and this is a principle in science that we call parsimony. So, how do we know that the current SARS-CoV-2 virus spilled over from bats? Right? So this virus was not created in the lab. It was very likely found originally in bats. And we know this because we've taken genetic sequences from bats all around the world, and we've found a number of viruses in bats that are 98% identical to the SARS-CoV-2 virus that spilled over into humans. So even though we haven't found the exact same virus, all of these viruses in bats are extremely similar. And you'll note that all of these viruses that are extremely similar to SARS-CoV-2 were found in bats in Zhejiang, China, which is not too far from Hubei province. So here are some questions that you should ask when you're reading and looking at the phylogenetic trees in this paper. So what genes did the authors use to compare the different virus strains in their study? What are the functions of these genes for the virus? And would you expect these genes to be conserved or similar in all the different viruses? And why or why not? For each figure where the authors compare the nucleic acid or amino acid sequences, make sure you know whether they're comparing a nucleic acid sequence, RNA or DNA, or an amino acid sequence. Then, how did the scientists get the data on the sequences that they are comparing for each virus? The last thing you'll want to know in reading this paper is a technique called immunofluorescence microscopy. So this is the basis of a number of important experiments that they show in the paper. So this technique is called immuno because it uses antibodies. And antibodies are products of mammalian immune systems. And in the immune system, their purpose is to recognize or tag pathogens. The way they do that is that the immune system generates an antibody for each pathogen that comes into the body. And those antibodies stick to the pathogens by binding very tightly to a molecule that's only found on the pathogen. You can see the process that antibodies go through in order to tag pathogens in the figure at the bottom. So these Y-shaped proteins stick on to pathogens and then either inactivate those pathogens directly by globbing them up or by signaling to other immune cells that this is a pathogen to be engulfed and destroyed. So antibodies are really, really good at sticking to molecules. And those molecules on the pathogen that those antibodies stick to are called antigens. They stick to those antigens by binding very tightly to a specific sequence in the protein that they're sticking to, and we call that specific little piece of the antigen an epitope. So because antibodies are really, really good at sticking to molecules, especially proteins, we can use antibodies to identify molecules in cells. So molecules are generally way too small to see, but antibodies are really, really good at globbing onto them. So we can generate an antibody that attaches to a specific molecule by provoking an immune response in a laboratory mammal, such as a mouse or a rabbit. Then we can extract antibodies that are really, really sticky for a specific molecule. We then can make those antibodies visible by attaching a fluorescent chemical to them. Right? So a fluorescent chemical is a chemical that emits light when it is excited by another wavelength of light. Right? So usually a laser will excite the fluorophore, and then it will emit light on its own. Right? So the antibody can stick to a specific molecule, and so if the molecule is present, the antibody will stick to it. And then the fluorophore lights up that antibody so that you can see it. It's bright enough to see with a microscope. Right? So we can use this technique to perform microscopy as is shown in this diagram here. So we have a immunofluorescence microscope, and there is an excitation laser that specifically induces that fluorophore to emit light. When the laser shines on the antibody with a fluorophore, then it will shine back light to the detector or to the eyepiece. So we can build immunofluorescence microscopes that have several different lasers so that you can excite different fluorescence molecules in one single image. For example, in the image on the right, we have 3 different pictures that were taken with 3 different excitation lasers. Right? So the one that has been colored blue was excited with wavelength 457. The one that is colored green was excited with wavelength 528. And then the one that is shown in red was excited with wavelength 617. Right? So you can take all 3 of these pictures by simply looking at the same cell and shining a different laser at it. And since it's all the same exact spot on the cell, just excited with 3 different lasers, you can overlay those 3 different images on top of each other to show where these 3 different molecules that were all detected by these 3 different antibodies are present in a cell. And so you can get this really beautiful picture of some of the proteins that are involved in mitosis, as is shown on this image here at the bottom. So it's important to note that fluorescent antibodies aren't the only things that we use in immunofluorescence microscopy. Sometimes we will use fluorescent chemicals themselves, specifically DAPI, which is a fluorescent chemical that also tends to intercalate or stick between the DNA bases and DNA double helixes. Right? So if you stain cells with this DAPI chemical, the DAPI will stick to areas of the cell where there's a high concentration of DNA, and then you can get a strong fluorescent signal from those areas that have a high concentration of DNA. And so you should know where would you find a high concentration of DNA inside of a mammalian cell. Right? So I'll ask you that in class. You can use DAPI to identify those areas of high DNA concentration as an additional marker in your immunofluorescence images. So think about these questions when you're looking at those immunofluorescence experiments in this paper. What molecule or antigen are the scientists trying to detect with the microscope images? And what does it mean when the RNA-dependent RNA polymerase antigen is co-localized within a cell?